time. And thank you so much for coming over. This is the second Hanover seminar this semester. And it's my honor to introduce Dr. Andy Cortese. And so he is the postdoc at the plant science department at the Dr. Black's lab. And I met Andy first time at the Forest Ecology Journal Club that I have been organizing. And I was so impressed that he gave really good thoughtful comments to the journals, and he led the discussion super well. And I just sent him the invitation for Hanover seminar, and he accepted my invitation nicely. And I also find that he's a fisherman, so I sent him a bunch of fishing trip, invitation, but he never accepted them. I think he was intimidated by the great fisherman. Come on, it's a fisherman job. He is supposed to laugh at this one.
No laughing at the truth.
No, it's not true. Okay, so Andy's gonna make up for my poorly received joke today by talking about the ectomycorrhizal fungi and make this seminar great again. And it's all yours. Okay.
Thank you for the introduction, Aki. So I'm Andy, and I'm going to talk about ectomycorrhizal fungi in mixed forests comprised of ectomycorrhizal and arbuscular mycorrhizal trees. A lot of this will be pertaining to my dissertation research, in which I was looking at the seedling establishment of lots of historically dominant ectomycorrhizal trees, as well as some of the current work I have coming here at MSU. Missing something? Oh, sweet.
Okay, that's good.
Okay, so some of you might wonder what mycorrhizae really are, and it translates to fungus root from a combination of Greek and Latin roots. So myco meaning fungus, rhiza meaning root. And it's usually a mutualistic symbiosis between plant roots and fungi, not always mutualistic. There's some really cool exceptions to this. But in general, you have a photosynthetic plant here, like this oak tree, nice green leaves, fixing sugars, and transferring a large portion of those sugars down to the root systems and associated mycorrhizal fungi, which in turn grow into the soil from the root system and helping with the acquisition of mineral nutrients and water. So the fungus is receiving its carbon source, only source of carbon, and the plant is receiving nutrients and water that they both need to grow. So usually both partners benefit. There's really two dominant types globally and several other really interesting types that I would be happy to talk about, just not in this talk. First being arbuscular mycorrhizae, or AM. And so a really good example of an arbuscular mycorrhizal tree in the northern forest is the genus Acer, or maples. Very, very dominant in many of the forests here. But actually about 70% of all plant species associate with arbuscular mycorrhizal fungi, so the vast majority of them. In contrast, the other main type is ectomycorrhizae, or EM, as you'll see abbreviated. On a few figures, you'll see ECM, means the same thing. So really, like, think of oak trees and other members of the Fagaceae, and lots of really dominant high latitude tree species. So although only 5% of all plant species, give or take, form ectomycorrhizae, they comprise something like 60% of all tree stems globally. So they can be very dominant. To take a closer look at an arbuscular mycorrhiza, they are formed by fungi in the phylum Glomeromycota, very ancient. They've been around for at least 440 million years. And they're thought to have helped the first plants colonize land based off of fossil records from the Devonian. Only about 1,000 species of fungi. Of course, what we define as a species for AM fungi is a little bit confusing as their taxonomy is kind of messy. but they actually have no known sexual stage, yet they maintain really incredibly high genetic diversity, which is confusing. So they actually produce these asexual spores, which for a fungal spore are massive. They can be up to 300 microns in diameter, so like a third of a millimeter. So for a spore, that's huge. And these just grow outside of a colonized root like this, just out in the soil where they eventually get moved around. Again, most plants associate with arbuscular mycorrhizal fungi. So just in terms of that ratio of fungal to plant diversity, it's very skewed towards the plant diversity side. Again, really important to think of dominant trees, so acer or maples. One important thing to note about arbuscular mycorrhizae are that the fungal hyphae actually penetrate the plant root cell and then a root cell wall, whereas other mycorrhizal types may not do that. Additionally, they offer no protective barrier along the root, which will come into more relevance later. So again, think of maple, also many herbaceous plants such as goldenrods, which are really dominant old field species following agricultural land abandonment. However, these fungi really have a limited ability to access organic forms of nitrogen, so they largely proliferate in areas with high amounts of mineral nitrogen. So ectomycorrhizal, or EM, fungi in the phyla Ascomycota or Deciduomycota, or actually both phyla, way higher diversity. And these actually evolved around 60 or 70 times from saprotrophic fungi. And also many of these fungi produce colorful fruit bodies, so they're macrofungi. So Deciduomycota produce mushrooms, among other reproductive structures. Ascomycota, think of truffles and other cup fungi, as well as lots of really just inconspicuous fruit bodies like crusts fruiting in leaf litter. Again, they primarily associate with woody plants. And again, that ratio of fungal to plant diversity is much higher than for arbuscular mycorrhizae. And when you look at an arbuscular mycorrhizal root system, one thing that's really cool is that you can actually see the fungal structures, sometimes with a naked eye, like on this white pine seedling. And so there's a cluster of colonized root tips right there. And so that color that you're seeing, that cream color, is actual fungal mycelium covering the root tip, forming what's known as a mantle. And so if we're to zoom in on this root, you can really see that this entire surface is actually isolated from the soil matrix by this fungal mycelium, which has really interesting implications for how the fungus can exert a lot of control over the plant in terms of nutrient uptake. Of course, you take this root and you take a salami slice off of it and make a mount underneath the compound microscope, stain it so all the fungal structures turn blue, and then you can get a really good look at the inside of a root tip. This blue is the fungal mantle covering, again, the entire exterior of the root tip. And then in between the root cell walls, they do not penetrate the root cell walls in an ectomycorrhizae, is the Hardig net, which is where nutrient transfer takes place. So it's thought that this morphology of ectomycorrhizae enables some degree of pathogen protection for ectomycorrhizal plants, just from that actual physical barrier with the soil. Additionally, these fungi, being that they evolved numerous times from saprotrophic fungi, have a degree of retained traits that enable them to access organic nitrogen. And so they're really thought to be highly functionally diverse. Here's kind of a bad picture, but here's a root tip that's totally smooth with no hyphae or mycelium growing out into the soil. And there's varying degrees of increasing amounts of fungal mycelium that are produced. This enables greater exploration in the soil for nutrients, as well as sometimes retained enzymes that can get nitrogen from recalcitrant sources of organic matter, such as conifer leaf litter that decomposes very slowly. So now I'm really focusing on ectomycorrhizae now. That's my area of expertise. So it's important to think about how plants and fungi in an ectomycorrhizal symbiosis are intimately linked. Really, they co-evolved over time. And so there's some degree of host specificity by which a given ectomycorrhizal fungus will have a range of host ectomycorrhizal plants from which they're compatible with and can associate with. And this usually occurs at the plant family level, but there are some interesting exceptions, which I'll talk about in just a moment. So here's an ectomycorrhizal fungus you may have seen around, the painted swillus. Suillus spraguei, very colorful. So there's one example of an ectomycorrhizal fungus. And then we have Inoculum geophyllum, which does not produce any fruit body that we know of, only asexual. So I'm just showing some roots here. And so Suillus spraguei only associates with eastern white pine, while Ectomycorrhizal geophyllum associates with just about every known ectomycorrhizal plant. And so in thinking about the host range of these fungi, it can go from extremely narrow, being a host specialist, like the Suillus, or broad, like a host generalist, like Ectomycorrhizal geophyllum. And so this specificity phenomena can influence the composition of ectomycorrhizal fungi, dependent on the composition of host trees above ground. And so this can ultimately lead to increasing ectomycorrhizal fungal diversity with increasing ectomycorrhizal host plant diversity, which has been shown generally on a site-by-site level. Okay, so many of you have probably heard of mycorrhizal networks. It's been a pretty hot topic in the kind of popular science media even lately. And so it's really just the linkage of a minimum of 2 plants below ground by the mycelium of one ectomycorrhizal fungus, in this case of some Scots pine seedlings right there. And so these can become really complex with multiple species of ectomycorrhizal fungi linking multiple species of host trees below ground, which has some really neat implications for how they might influence above ground community and competitive dynamics between trees. And so if you have a little seedling establishing here, that may be a lot more going on than just competition for light above ground. However, this is controversial. Some people may have heard of the mother tree hypothesis, and this is based off of some experiments largely in the 1990s by which they showed carbon transfer below ground from an established plant to a shaded seedling, which got a lot of traction in the media in terms of thinking of plants helping each other below ground. But this kind of violates a lot of ecological and evolutionary theory. However, again, it got a lot of attention, getting the cover of Nature back in 1997 with the Wood Wide Web. It's a pretty kitschy name. However, there's been a lot of pushback and a lot of over-interpretation of what ultimately were statistically significant, but not necessarily ecologically significant results. Happy to elaborate more on that later. Really, it's kind of become more accepted that this whole idea of altruism below ground doesn't really exist. That said, these mycorrhizal networks can still be very important in terms of tree seedling establishment. And so really just going to go back to the 1980s with this great paper by E.I. Newman, just before any of these thoughts of, really like intentional benefit to host seedlings came to light. And so there are some realized benefits that these mycorrhizal networks can really provide for an establishing tree seedling. First being that having this active ectomycorrhizal fungal inoculum, or mycelium rather, is a very effective inoculum source for, in terms of rapid colonization of a tree seedling's root systems. So this fungus, remember, is getting its only source of carbon is from the photosynthetic products of these seedlings here. And so being that it's kind of connected to a carbon source, it can proliferate and grow pretty rapidly in the soil and then actually colonize the roots of this newly planted hemlock seedling much faster than if it was from the germination of dormant propagules and spores, which would mean that the only source of carbon for those fungi would actually be from this establishing seedling. Think of a forest, low light environment, small seedling, probably going to be happening a lot more slowly. Additionally, all that investment into this fungal mycelium by these established pine seedlings represents a huge carbon subsidy, knowing that these fungi are heterotrophs and completely dependent on the host plants for their carbon source. So now there's all this fungal mycelium in here that this seedling didn't really have to pay for the construction of, right? And so this can also enable greater access to soil nutrient pools and have a larger zone of interaction with the soil. And also can lead to greater fungal richness on seedlings as well, just through some of the biotic filters, kind of preventing the germination of fungal spores, leading to the successful establishment of an ectomycorrhiza. And so given all these things about mycorrhizal networks, it's really cool to take a step back and think about how scattered ectomycorrhizal patches within a non-host matrix might be really important for the maintenance of ectomycorrhizal plant species or even recolonization following a disturbance. And so there's some realized benefits of these networks for seedling establishment, such as right here on Mount Fuji in Japan, from Nara, did some wonderful work in primary succession out there. And kind of a bad picture, but these light green patches are actually a dwarf willow, which is more of a facultative ectomycorrhizal plant. They can grow independently of the fungi for at least part of their life cycle, growing in volcanic ash on Mount Fuji. And you only see ectomycorrhizal, later successional ectomycorrhizal plants establishing near these patches. And even non-ectomycorrhizal vegetation would not have these seedlings in it. So it goes beyond just abiotic effects. So ultimately, these patches can be associated with a higher abundance and diversity of ectomycorrhizal fungi and these mycorrhizal network access potential, much more than that surrounding matrix. And also thinking in terms of plant soil feedback too, these ectomycorrhizal plants can really kind of provide those benefits. So this has been shown mostly under primary and secondary succession, where we have enhanced seedling survival and growth near these patches as opposed to far away from these patches. However, this has not been really that well studied in northeastern North America. Much of this work actually came out from my major professor's research out in California following fire, as well as other parts of the world. But one important thing about mycorrhizal patches is thinking about how fungi might be able to disperse from one of these patches out into this non-host matrix, and then provide fungal inoculum for any tree seedlings that may be landing and seeds that are germinating far from these ectomycorrhizal patches. And so actually a lot of these fungi might be dispersally limited based off of the limited data we were able to collect on their fungal dispersal. But when you see a mushroom such as this frost bolete right here, which produces wind-dispersed spores, these fungal spores are tiny. You're talking in the order of like 10 to 15 microns across. So you think that when the wind blows, they're going to go really, really far. Like most of them are just going to go meters and meters from the cap of a mushroom. But in reality, about 95% of all the spores from a given ectomycorrhizal fungus, they land 25 centimeters from the cap of that mushrooms. They don't go very far. That still means there's probably very few at this tail end of the spores that do get into turbulent air vortices and go far, but it's going to be pretty diffuse and variable over space and time. So taking a further step back and really thinking about how these AM and EM patches might work in eastern deciduous forests. Of course, really talking about this region, more of the northern hardwoods, white pine, hemlock forest region in upstate New York and then into New England. So it's a mixture of AM and EM trees, where there's really cool research from my lab's current collaborator, Rich Phillips, on the nutrient cycling dynamics of these forest patches. In general, the AM forest patches have really fast nitrogen cycling. This is largely due to their leaf litter decomposing more fast. And so generally with greater pools of inorganic nitrogen and just greater litter turnover. Whereas the EM patches tend to have more recalcitrant leaf litter, so it degrades much more slowly. And these tend to have much slower nitrogen cycling, generally dominated by organic forms of nitrogen. However, due to a lot of really human impacts, there's been a pretty large increase in the abundance of AM trees and a decrease in EM trees compared to the pre-settlement forest. And this is due to lots of things such as nitrogen deposition, where a lot of that nitrogen is actually coming from industry here in the Midwest, getting blown over and being deposited as nitrate, which is pretty much the most plant available form of nitrogen. Also fire suppression, as lots of the indigenous people used fire to manage the landscape, which hence has been suppressed. Selective logging, where trees such as white pine were really highly valued and have been logged out of many forests. And of course, pest and disease invasions as well. But one of the biggest impacts is actually through agricultural land use. And so about half of the forested area in the Northeast is actually forests that have regenerated on abandoned farm fields. And they're really characterized by having a lack of those spring ephemeral wildflowers, which is what makes the eastern forest really, really neat. In the spring, you get all these cool plants like trillium or blue cohosh. These are largely absent from these forests. They have not managed to recolonize them. So in thinking about the succession of a forest, this is like agricultural land use. These forests that have developed were in this stage over here, well past canopy closure, very different tree composition than that pre-settlement forest. And so here's a picture of one of my study sites in I would say pretty much a textbook secondary forest. One thing you'll notice is the stone wall here, right? So that was probably from somebody who was tilling that field and got tired of hitting rocks with their plow. They stacked them in all these nice stone walls that I eventually tripped over with transect tapes when I was doing some tree surveys here. Also notice the forest floor is almost completely flat with no pit and mound topography, directly a legacy from agricultural land use. And then if you look at the trees, you probably can't tell from here, but this is almost exclusively one age class of sugar maple, which those trees in particular respond really well to just old field succession, and they've been very, very competitive. And so what we found is that in this region, there are lots of forests dominated by AM trees with just very few ectomycorrhizal trees present, which implies really diminished ecosystem services and other benefits compared to the pre-settlement forest condition. And of course, I'm always curious, what about the EM fungi? And especially when I think about residual forests. So these are forests that have never been cleared for agriculture. They could be an old growth forest or a virgin forest that has never been cut, or even one that was maintained as a woodlot throughout time, logged repeatedly, but has never been tilled and converted into a farm field. So in general, these forests have more biological legacies than a secondary forest. And if you look here, here's an example of part of an old growth forest at one of my study sites. Right in the front is a mature eastern hemlock tree, which in particular are very, have a really poor ability to colonize these post-ag forests. Multiple age classes of trees here comprised of hemlock, beech, also some maple and other AM trees present there. If you look at the forest floor, there's some pit and mound topography and a pretty robust herb layer dominated by various ferns. And so with all these biological legacies here, again, what's happening below ground that we cannot see, especially because these forests exist usually as small patches surrounded by secondary forest. So it makes me wonder if these might be reservoirs of ectomycorrhizal fungi, kind of as a source where they might be able to disperse into the secondary forests. And this brings me to my first research question, which I wanted to look at ectomycorrhizal fungal communities along a distance gradient from the edge of a relict ectomycorrhizal tree-dominated old growth forest going out into a secondary forest dominated by maple and other AM trees. Then I wanted to see how tree seedling physiology was also influenced by this distance gradient from that old growth forest edge. So right here is two pictures of the same exact area. So one is from 1926, an aerial photograph. The other one is Google Earth imagery from a few years ago. And of course, this is at my study site north of New York City at the Mianus River Gorge Preserve. And I lined these up just by tracing some roads. So it took me way too long to do this, but then I was actually able to match up the spatial extent of each of these maps. And hopefully the first thing you'll notice, other than the difference in color, is that much of the area in 1926 existed as a matrix of agricultural fields and pastures with just a single row of trees kind of separating them, where much of that area is now forested now. So this is an example of a secondary forest here. What you may also notice is that there's a steep river gorge here, which was clearly difficult to log because it was never logged or cleared. And that is an old growth forest dominated largely by ectomycorrhizal trees such as eastern hemlock. And so for this study, I decided to run transects out from the edge of that old growth forest, going just 200 meters into the secondary forest. And at each point here, I measured tree composition and collected soils to then take back to the lab and grow ectomycorrhizal tree seedlings in. And when looking at the tree composition though, going from that edge, if you look at the blue line, just ectomycorrhizal tree basal area, it drops right off. So after about 100 meters, it's almost devoid of ectomycorrhizal trees in this forest. So there's some pictures from the field. There is my former intern, Allison, helping measure trees while her mom, who's nice enough to come out, is bleaching a shovel in between samples. Of course, we wanted to keep things sterile. And this is at the height of COVID, so we wore masks outside. Seems like such a long time ago. So we collected soils largely from the upper 15 centimeters of the soil, which is where most ectomycorrhizal fungal activity should be in the system, and brought them back to SUNY ESF, where my lab was. And then I grew black birch seedlings in a growth chamber bioassay. Grew them for 7 months, and I took some physiological responses, including gas exchange measurements from them prior to harvest. I then harvested all of my seedlings, cleaned off their roots, and then I scored ectomycorrhizal colonization visually using just under a dissecting microscope. Then I also sorted ectomycorrhizal root tips based off their color, branching pattern, and presence of any extra radical hyphae or mycelium growing out from that root tip. I then did some old but still usable molecular methods to identify each unique morphotype per seedling to an equivalent of a species level using restriction fragment length polymorphism and Sanger sequencing. So what I found was, if you look at the top panel, this is OTU richness per seedling, again, a proxy for species in the microbiology world. And on the bottom is the proportion of ectomycorrhizal root tips, 0 being totally uncolonized, 1 meaning every root tip was colonized. And on the left is just showing that relationship with distance from the old growth forest in meters. And on the right is from the most parsimonious model from AIC-based model selection. And so looking here, we could see a decrease in OTU richness and proportion of EM root tips as we moved away from that old growth forest edge. With the root tips, it's in particular a drop off after 100 meters with that drop off in ectomycorrhizal trees. But looking at the most parsimonious models that we identified for OTU richness here was actually an interaction between EM tree basal area and distance from the old growth forest edge. So at each distance, we found positive effects of EM tree basal area on the OTU richness per seedling. But then when looking at these y-intercepts at that old growth forest edge, even if no ectomycorrhizal trees were in that immediate vicinity, they still had at least 1 OTU per seedling on average. But those grown really far from that old growth forest edge had much less than 1 OTU per seedling on average, indicating really low diversity. When looking here for proportion of EM root tips, we found that it was best explained actually by the proportion of ectomycorrhizal trees in each neighborhood. I'm showing some effect of just these established trees on kind of propagating these ectomycorrhizal fungal propagules in the soils. We also looked at community composition with a redundancy analysis with colors corresponding to each distance from the old growth forest edge. And we found actually that ectomycorrhizal tree basal area here was the most important predictor for community composition. So kind of moving out from this old growth forest edge, the community becomes much more homogeneous along this axis, also corresponding to decreases in ectomycorrhizal tree basal area. And so what these results really suggest is that ectomycorrhizal fungal diversity is really influenced by the dominance of ectomycorrhizal trees in these mixed arbuscular ectomycorrhizal forests. And so I spent more time than I'd like to admit probing for any physiological response of these black birch seedlings to fungal colonization, soil conditions, even tree composition. And really the only result that stuck out was a negative relationship between the photosynthetic rate per unit leaf mass with distance from the old growth forest edge. So a pretty modest decline, but for almost everything else, we found basically no effect, which I think might relate in part to some of the neat traits of black birch, which I'd love to talk about with anyone later with some ideas. So this leads me right to my next suite of research questions from separate chapters of my dissertation. And so I was really curious seeing at this effect of EM tree dominance on ectomycorrhizal fungal diversity in these forests to focus on those scattered ectomycorrhizal tree patches in this matrix of secondary forest. So I want to see the influence of these patches on fungal colonization of seedlings, as well as the fungal community composition, as well as the physiological response of seedlings as measured for their survival and growth. Then I also wanted to test a soil inoculation experiment, taking soil from a residual forest, such as that old growth forest, and moving it into the secondary forest as a potential management tool or restoration technique to reintroduce ectomycorrhizal fungi where they may be missing. So again, looking at inoculation effects on both the EM colonization of seedlings, as well as the physiological response of them. And so I used the same site actually for part of my study here at the Mianus River Gorge, again, focused on these areas of secondary forest, again, largely dominated by arbuscular mycorrhizal trees. So really thinking of these areas here, just extremely low ectomycorrhizal tree abundance. And I did this in both a growth chamber and field. And for the field component, I added 2 additional sites in central New York. So my basic experimental design for both the growth chamber and field experiment involved identifying focal AM acer or maple trees and focal EM or betula trees at each site. I then pretty much utilized a control where I planted tree seedlings directly next to those focal trees or next to those focal trees with the addition of EM forest soil inoculum. And of course, this also included collection of soil and setting up an experiment in a growth chamber. And so for this work, I really carried lots of buckets through the woods. Sounds really glorious, but my arm got a good workout, so that was cool. Excuse me. And so again, I used this design as part of a multi-year field experiment where I planted Tsuga canadensis or eastern hemlock, eastern white pine or Pinus strobus, and multiple species of oaks, just due to availability of acorns as their masked species. And I established paired subplots next to each of my 40 focal trees that I had selected, where one subplot was a control and the other one received that EM forest soil inoculum. And of course, when you have an experiment going in the woods for a few years, some unexpected things happen, such as trees falling on my subplots, or another plot, I had a tree land on one subplot, and then I actually launched a stone from a stone wall onto the other subplot, but only killed like most of the seedlings. So I still got data out of it. And then I also set up a growth chamber experiment just with hemlock and pine. I had issues with oaks for this one, which I only grew them for 9 months. And this picture is just to highlight that I was also a TA when setting this up, and I had a weekend to plant one site for this and get the soils for this. So we had to work into the night pretty extensively. So here's some results from the field component. So right here is EM colonization, this time as a percent on the y-axis, and then all of my treatments here on the x-axis. And so just looking at Tsuga, or the hemlock seedlings, notice that seedlings planted next to the AM acer, or maple tree alone, had extremely low EM colonization compared to those planted next to the ectomycorrhizal betula or birch trees. And this was consistent for all 3 genera of seedlings that I planted, but most pronounced for hemlock. And when looking at the addition of EM forest soil inoculum here, it only increased the colonization of roots for seedlings planted next to the AM acer tree, and it had no measurable effect on seedlings planted next to the EM betula tree. And again, showing OTU richness as a proxy for species. Similar trends here with really low richness on seedlings planted near the AM acer tree, much higher near the EM betula, but we found a little bit less of a response for inoculation, where it just made these mean kind of similar to somewhere in between maple and birch alone, but no effect at all for seedlings planted next to the EM betula trees. And then when looking at just the diversity among all of my treatment combinations, we found that there was much lower overall fungal diversity for seedlings planted next to the AM acer trees. This is a species accumulation curve. So it's starting to saturate even after about 20 subplots at around, I think, 23 ectomycorrhizal fungi that I found. In contrast, look at the blue line, which is for seedlings planted next to the EM betula trees, and much higher diversity. I think I found 63 ectomycorrhizal fungi, and this isn't even close to saturating, indicating that with more added subplots and more sampling, I would have continued to detect new ectomycorrhizal fungi. When looking at the effect of inoculation, however, it again was only effective at increasing the number of detected ectomycorrhizal fungi for seedlings planted next to the AM acer tree. And finally, we looked at fungal community composition with a non-metric multidimensional scaling. Colors are the same as before. So the red here is the community for seedlings planted next to the AM acer. And notice it's really distinct from the other treatments. And also the ellipse is much smaller, indicating a more homogeneous community. If you look at the blue, that's for birch again. Notice that it's more heterogeneous and it doesn't overlap all the way, showing some differences in composition. But again, when looking at inoculation, if you look at this orange ellipse, it's incredibly different from the community associated with AM acer, indicating a really great effectiveness of this inoculum in altering the fungal community in areas lacking ectomycorrhizal trees. And of course, this discrepancy was detected through an interaction term that was significant in a permanova using the same distance matrix. Okay, so now we're going to jump to the growth chamber part, where again, we only group pine and hemlock, did not grow oak, and I apologize for jumping to common names here, but different publication. So what we found for hemlock or Tsuga seedlings was those planted next to the EM betula trees grew about twice as large than those planted next to the AM acer trees. In general, they also just looked much healthier. But for both soil types, we found a dramatic increase in biomass with the addition of that EM forest soil inoculum. And so there's an example. Here's a hemlock grown right in that maple soil. They mostly looked like this, like really stunted. They're really, really chlorotic and just, they pretty much just stopped growing at right around that size. Whereas those either planted in birch soil alone or treated with that inoculum were much healthier, and they continued to put out new growth towards the end of the experiment. One interesting side note is I did do some nutrient analysis on these seedlings and found that this hemlock was the ones in maple soil alone were incredibly high in nitrogen, but I don't really understand why. So I'd love to, again, chat with people about that later. And then when looking at pine, we found similar trends, but they just seemed a little bit less responsive to being grown in birch soil. So they were not significantly different in size from those grown in maple soil alone. So really to kind of summarize what we found here was that these EM betula trees were associated with much greater diversity and abundance of ectomycorrhizal fungi compared to the surrounding maple forest. So these are really functioning as EM tree islands, really kind of similar to those patches kind of following disturbance that a lot of this research has been, founded upon more in the Western US and in Japan and Mount Fuji. Also found evidence for mycorrhizal network interactions, although we didn't specifically test for this. So really it was the composition of fungi that we detected from these seedlings. Again, really ones that do not effectively colonize the roots of these small tree seedlings from spore germinants. And this includes Cortese, which is very colorful, really neat mushrooms, also Amanita, and then Russula. So ones that really known from a lot of research will not colonize seedlings in a growth chamber bioassay condition. Also many other species too. Here's one of my subplots near a birch at one of my sites in central New York. Here's a Lacaria that was fruiting in the subplot I have in my hand here. And if you look at these brown structures, those are earth balls, so other ectomycorrhizal fungi. They're fruiting in the subplot. I'm assuming that was from the established ectomycorrhizal birch tree and not from the seedlings. Also found seedling growth responses. So Pinus and Tsuga growth was greater, both in the field as well as in the growth chamber. Although there's some interesting things about the plot setup where I think my growth responses were somewhat muted because the light environment was actually slightly shadier next to the birch trees. But again, these results are most clear from the growth chamber under really controlled conditions. So in contrast, near these AM acer trees, we really found few ectomycorrhizal fungi, really representing a depauperate community dominated largely by spore bank fungi. These are the ones that kind of produce like a soil seed bank of just resistant spores that can persist for decades in the soil. Some genera like Swillis, it's thought they can be viable for over 100 years in soil. So really important for post-disturbance ecology. And this includes a truffle-like fungus, Hymenagaster. This one's actually a basidiomycete, so convergent evolution, right, in terms of fruit bodies. A true truffle right here, so Ascomycota. And then Swillis on just the pines, because Swillis are specific to pines and larch primarily. And what's also interesting is that these fungi are all pretty well established to be eaten by animals and actually vectored as kind of like endozoochory. So an animal eats this mushroom here, such as this flying squirrel here eating a little truffle-like fruit body. Then they go and they move around and then they relieve themselves and ultimately releasing like large pockets of fungal inoculum that can form a spore bank in the soil. Additionally, lots of these common ectomycorrhizal fungi you would expect to find everywhere, they were really infrequently found these areas away from ectomycorrhizal trees. And this includes that Cenococcum geophyllum, really is thought to associate with about any ectomycorrhizal plant. Most papers will describe it as being ubiquitous, but it was really infrequently found far away from ectomycorrhizal trees. However, we found evidence that soil inoculum, is really effective in introducing some of these fungi into these areas lacking those native ectomycorrhizal fungi. So we found positive effects on ectomycorrhizal fungal colonization and richness near these AM acer trees, as well as enhanced growth of all seedlings. So we found an effect on the quercus seedlings in the field, a weak positive effect. And then also in the growth chamber, we found positive effects on growth for the pine and hemlock. And so from this project, what we really found is that there's few ectomycorrhizal fungi away from these ectomycorrhizal trees, really suggesting that expansive areas of secondary forest really have depauperate fungal communities, which has ramifications for the reestablishment of these obligate ectomycorrhizal associating trees that are largely absent. Additionally, we found evidence that greater EM dominance is associated with greater ectomycorrhizal fungal diversity, suggesting perhaps an EM dominance hypothesis, where fungal diversity in these mixed AM-EM forests is largely driven by the just basal area or the dominance in a given area by ectomycorrhizal trees, which we'll talk about a little bit more later. Also, again, found that a lack of ectomycorrhizal fungi can hinder the establishment of ectomycorrhizal trees. This, of course, is including a bunch of other factors like competition from maple, seed dispersal of the trees, lack of suitable disturbance. But it does suggest that this lack of ectomycorrhizal fungi in these forests really represents cryptic land use legacies, similar to that lack of those spring ephemeral wildflowers from these forests. And so when trying to reestablish ectomycorrhizal trees, you really need to factor in below ground associations to enhance your success, whether that means retaining any ectomycorrhizal trees if you're doing a silvicultural prescription to maintain those populations of ectomycorrhizal fungi. Remember, they will not grow unless there's compatible host trees present. So if you remove all the trees, you're going to knock those fungi back pretty hard. Excuse me. Additionally, use local EM forest soil as an inoculum. It's better than the commercial stuff, but there is the potential you will move pathogens with that beneficial fungi, of course. So more research in this is really needed. And so this really makes me wonder now, and what leads me up to my current work here at Michigan State, thinking about what are more of the patterns driving ectomycorrhizal fungal diversity in these mixed AM-EM forests. So digging a little deeper now. So we have two competing hypotheses I kind of hinted at earlier, kind of describing this relationships of plant communities with ectomycorrhizal fungal diversity. First being that ectomycorrhizal tree diversity should have a positive effect on ectomycorrhizal fungal diversity, and that's from some of the work I showed earlier in this presentation. Also, this idea of EM tree dominance having a positive effect on ectomycorrhizal fungal diversity. And of course, I'm incorporating some work from my colleague, Andrew Eager, who also came to some similar kind of results from some of his own work. Also, there could be soil nutrient effects on these ectomycorrhizal fungi. Again, thinking of these AM, which is associated with high rates of nitrogen cycling and more inorganic nitrogen, versus EM characteristics of more slow nitrogen cycling. So I didn't really touch on this, but many of these fungi are really susceptible to atmospheric nitrogen deposition, where it seems like they can be selected against due to their really carbon demanding growth strategies that are necessary to access those really recalcitrant forms of nitrogen. If you're getting a bunch of nitrate dumped on you, then those life strategies probably won't be as competitive. Of course, there can be some really weird non-linear responses when you have a bunch of recalcitrant and labile leaf litter mixed together, which may synergize and be completely unexpected, like slowing some of the cycling of labile components, such as from this recent paper from 2020. And also that leads me to wonder how AM trees might influence ectomycorrhizal fungi, including community spillover effects, where if you have a forest that's largely arbuscular mycorrhizal tree dominated, then perhaps the fungi and biogeochemical traits associated with the AM tree community might be exerting a really strong influence on ectomycorrhizal fungi and influence their composition. So again, thinking about how AM leaf litter, how that might affect these ectomycorrhizal fungi hasn't really been investigated very thoroughly. And so I decided to look at some tree ectomycorrhizal fungal diversity relationships, taking a second look at a lab data set. Again, this basically the data used for Andrew's paper, Soil Biology and Biochemistry, from just 2 years ago now. So I was really curious to see how ectomycorrhizal fungi are influenced by tree diversity. And this includes not only the diversity of ectomycorrhizal trees, as well as the diversity of AM trees and that entire tree community of combined AM and EM trees. I also wanted to look at EM tree dominance. So just the basal area per plot, whether a total or a relative measure of all the ectomycorrhizal trees per plot. And then also soil conditions. As I had access to a really cool data set with all this stuff, felt pretty lucky. So I was hoping to really be able to predict the diversity of ectomycorrhizal fungi in these mixed AM-EM forests. And this leads me to my next set of research questions. So I just want to know how AM, EM, or this combined tree diversity influence ectomycorrhizal fungal communities. As again, these AM trees are largely ignored in these studies, and people tend to focus just on the diversity of EM host trees. Also want to see how EM dominance influences ectomycorrhizal fungi and look for any relationships between EM dominance and tree diversity, as those aren't necessarily proportional, as you could have a plot totally dominated by ectomycorrhizal trees that's just one species, or a plot with low EM tree dominance of a wide diversity of ectomycorrhizal tree species. So just including both of these in the same statistical model might yield some interesting insights. Then I also wanted to look at soil characteristics largely attributed to that AM nutrient economy versus the EM nutrient economy on ectomycorrhizal fungi. So really thinking about availability of soil nitrogen here. So higher near AM, lower near EM. And so really briefly here, for my approach, I established plots across a wide range of EM tree composition in the Adirondack Park in upstate New York, one of my favorite places on earth. So there are 3 sites established. This is near Lake George, Newcomb, and then Shingle Shanty Preserve, which I think is like the biggest peatland in definitely New York State, a really cool area there. Only spanning a range of 21 to 100% EM tree dominance. And I forgot to mention this earlier, but for my own dissertation work, I was looking at a range of around 0 to 18% EM tree dominance. So largely, the previous study was all pretty much AM dominated forests. Again, had access to a really cool soil data set from our collaborator, Kurt Smimo, who is a soil biogeochemist. So had lots of cool data on like nitrate and ammonium levels in soils, nitrification rate, and other neat stuff that you really think could be influencing these fungi on a really biogeochemical level. And then had, of course, a fungal sequencing data set from Illumina MySeq from both soil and roots, just from soil cores taken from each plot. I then ran a bunch of linear models and really an exploratory framework and used AICC to select the most parsimonious models, and then I model average responses. So right here are some effects plots from my modeling where I have soil and root on the left and right respectively. Hill number 2 on the top in orange, which is similar to, is the same as the inverse Simpson's diversity index, so weights by the abundance. So it's your diversity of like your abundant species, really. And on the bottom is hill 0, that's just richness of fungal genera in this case. So it was really interesting was that out of these 4 different measures of fungal diversity, in 3 of the cases, we found evidence or positive effects of some inclusion of AM tree diversity on the diversity of ectomycorrhizal fungi, completely unexpected by me. And this includes a phylogenetic signal here, just AM tree species richness, and then combined AM and EM inverse Simpson's diversity. And so also forgot to mention that if these error bars, these are 95% confidence intervals, if they overlap the 0 line, we considered it non-significant. If they don't overlap, then they're considered significant. So these 2 responses here were non-significant because they overlap 0. Then we're looking at, testing for EM dominance. We did find some evidence in support of that for both soil and roots, but only for hill number 0. I'm interpreting as having a great effect on the abundance of just richness, but then no effect on, again, that diversity of those abundant taxa. And then finally, when looking at soil nitrogen, thinking of more soil nitrogen is more associated with that AM nutrient economy. We found negative effects in 3 out of the 4 cases for some measure of soil nitrogen on fungal diversity. And so we also decided to go and look at every single measure of diversity altogether and see if these results kind of aligned with what I found from my other step of model selection. And so sorry, this is kind of cluttered, but AM diversity is in red, EM diversity is in blue, and then combined AM and EM is in purple. And so interpretation is the same. If these error bars don't overlap 0, they are significant, and the significant ones are bolded. And so what we found, again, was that the biggest effects on ectomycorrhizal fungal diversity was from some inclusion of arbuscular mycorrhizal tree diversity. Even here, where there's a significant response for ectomycorrhizal diversity, the AM diversity had a bit of a stronger effect. So found this pretty fascinating and also offers support for EM dominance and EM soil characteristics. Again, just viewing nitrogen as being more associated with that AM nutrient economy. However, we didn't find really much support for the EM diversity hypothesis. In fact, again, AM tree diversity appeared to have a really strong positive effect on ectomycorrhizal fungal diversity, completely unexpected by us. And so there's a few proposed mechanisms we're kind of formulating now, and we think there could be something to do with AM tree roots and below ground competition for nutrients, increasing the diversity of these fungi. So AM trees tend to really have prolific root growth, and they'll just actively grow their roots towards nutrient hotspots. And really, like, if anyone's ever had a garden bed next to a red or silver maple tree, and then like you go to dig it up and it's completely encased in roots, I think these trees can be very aggressive in proliferating the roots. So there could be some effect there. And then having greater diversity of these roots may enhance the overall uptake of these mineral forms of nitrogen, thereby actually resulting in a nutrient limitation potentially for ectomycorrhizal trees, therefore increasing the reliance on these trees for their ectomycorrhizal fungi to access those recalcitrant forms of nutrients. Also increased litter complexity, having greater diversity of AM and EM trees perhaps in the same plot, resulting in these sort of nonlinear responses in biogeochemistry, making conditions more favorable for ectomycorrhizal fungal diversity. And also just fungal competition. So AM trees are generally associated with more saprotrophic fungi than EM trees. And there could just be some interesting interguild interactions below ground between saprotrophic and ectomycorrhizal fungi. So this really highlights that there could be non-host interactions between these AM trees and ectomycorrhizal fungi, but we really don't know the mechanism driving this at this point. And so we really need to come up with some manipulative experiments to start testing this. So one could be just adding a bunch of AM leaf litter into ectomycorrhizal plots and seeing if that changes fungal community composition. Okay, and this went a lot faster than when I practiced it, so I do apologize. But I do want to thank so many people, especially Aki, for having me here today. Everyone in the Blackwood lab, my PhD committee, all the collaborators and wonderful people I've worked with here at MSU. Then finally, plant soil microbial sciences, plant biology, and then everyone in the forestry department for coming here and listening today.
Questions can take a few.
Yes.
Yeah, one thing I was wondering was if it's known, like if you have a tree that's standing dead or a tree that's been cut down, how long can the mycorrhizae live on the root system after? Like if you mentioned synvocal prescription, you come in and plant seedlings after the mature tree is cut down. Do you have like a buffer period where they can still get an
Absolutely, it's a great question. So what will likely happen, I don't know how long it would take for the fungal mycelium. So the active fungi, again, they need access to photosynthetic products from that tree to grow and survive. So those fungi will die at some point. I don't know when, but many of these fungi will produce resistant propagules. So this can include hyphal fragments, sclerotia, and also resistant spores that can also persist for very long periods of time under these dead trees. However, there's some evidence that these network interactions can have a much greater effect at just enabling the mycorrhizal colonization of planted seedlings. And remember that many ectomycorrhizal fungi can only colonize the roots of a seedling through mycelium, just through like the biotic barriers to spore germination and formation of ectomycorrhizals.
Nice.
Great talk. Thank you so much. I was wondering, do you know how invasive earthworms might influence the whole mycorrhizal?
That's such a great question. There was someone in my lab who did a master's thesis, my PhD lab, who did a master's thesis on it, but they never got any of their sequencing to work. So they don't really know how community composition was altered, but I think it must be dramatic. So in central New York, where I'm from, we've had in the past 10 years, the Asian jumping worms invade, and they completely alter the soil environment, and they turn everything into like granular, like coffee ground, and no discernible like organic and upper mineral layers. So it must be profound, but I don't know specifically off the top of my head. But great question though.
Okay, let's give another hand to Dr. Andrew.